Bismillahirrahmanirrahim. I welcome you all for the online class for the introduction to nervous system. Inshallah, you will have a very easy lecture and very informative lecture today.
Doctor, can you record the session, please?
Yes, recording has started.
All right.
I know most of you are not in a proper position, or some of them are just joining, some of them may be sleeping. So you can refer it later on, no problem. I will start sharing my screen now. Where is, okay, this is the share button. I will share the window. You can see my screen now.
Yes, doctor.
What's your name? Yeah, I have Omar with me offline. He is in front of me. Bismillahirrahmanirrahim. So today's lecture is about introducing yourself for nervous system. In our body, we divide the whole to study. We divide the things into different systems like cardiovascular system, like immune system, like urinary system or reproductive system or gastrointestinal tract, digestive system. So one of the system which is very important, all the systems are actually very important, but the one which is controlling the other systems is the nervous system. So in foundation block, we will be showing you how the arrangement of the nervous system is and what are its parts and what are the functions of each individual part. Okay, so at the end of this lecture, you should be able to describe the organization of the nervous system, how you divide central, peripheral, afferent, efferent, something like that, organizational. Then you should be able to describe the functional classes of the neurons. We have already discussed in different classes about the neuron, whether they are afferent neuron or efferent neuron or interneuron. So we will see how they are classified functionally on the basis of their function. Some are carrying the information, some are bringing the information, something like that. Then apart from nervous neurons, because you know every system are made up of specialized cells. Apart from that specialized cells, there are some supporting cells. Okay, so some supporting cells could be, it could be uh, uh, it could be glial cell, it could be astrocyte. So we will see it in nervous system. What are the supporting cells? What are the supporting cells? Then you know the nervous system is it's a very important organ or system. It has to be protected. So we have to discuss some special issues related to the protection of the nervous system, like the covering of the brain, like the covering of the spinal cord, and not only the covering physical, but there is something called as cerebrospinal fluid. Cerebrospinal fluid. Can I edit? CSF. Which short form is cerebrospinal fluid is nothing but CSF. As the name suggests, it is covering, it is a water-like substance, liquid, which is covering the brain and the spinal cord. The spinal cord. So it is around the spinal cord and it is around the brain. That's why it is called as cerebrospinal fluid. Okay. Let us move to the actual lecture now. So organizational system of the nervous system can be organized broadly into something which is called as central and another one is called as peripheral. So CNS is the short form of central nervous system, whereas PNS is the short form of peripheral nervous system. Central system and peripheral system. Why? Because of the position where it is placed, where it is placed in the center or in the body. It is placed in the center. That's why it is called as central, and something which is away from the center, we call it as peripheral. So we have got brain, the cerebrum, cerebellal cortex. Oh, sorry. It is look like this. Just the diagrammatic representation I'm drawing. And attached to the brain, there is a spinal cord, which is running through the vertebrae, reaching up till the pelvis, up till the pelvis. So this is all in the center. What is coming out? Let us say I will move it in the coronal section in front. If I move it in the front, it will look like this. Sorry, I'm drawing it very bad. Something like that. So this is central CNS, and out of this something is coming out. What are these? These are the peripheral nerves, which will be supplying the muscles of the arm, forearm, leg, or thigh. All these are called as the PNS, peripheral nervous system. Peripheral nervous system. So central nervous system, which is nothing but brain and spinal cord. Central is brain and spinal cord. Peripheral nervous system are the nerves which are coming out from the spinal cord and supplying to the organs. This is the diagrammatic representation of the nervous system. Here in the pink, the color which is in the pink, here you will see the picture, that is the brain, and attached to the brain down into the vertebral column is the spinal cord. The spinal cord and brain together are placed in a area we call it as central nervous system. They comprises of central nervous system. Through the central nervous system, the fibers are coming in and out of the central nervous system. We call them as peripheral nervous system. So yellow color area, this whole yellow color area is nothing but peripheral nervous system. Okay, all these are peripheral nervous system. Now in peripheral nervous system also we have got certain division. So what is the function of central nervous system? It is regulating the bodily activities. Regulating the bodily activities. What are the activities? What is the activities? In the form of temperature, pulse, thinking, memory, muscle or movement. All these are bodily activities. Apart from this, there are many. So it is controlled by brain and the spinal cord. The spinal cord is also independently can control movement also because of the local reflex. Whereas peripheral nervous system is divided into two. The nerves which are attached to the spinal cord, they can be bringing the information from an organ to the spinal cord and then getting it to the brain, taking it to the brain. So these are incoming signal. These are incoming signal. Incoming signal, we call it as afferent. Any signal, any information which is coming from the organ, or you can say from the receptor of any organ, then to the spinal cord and then to the brain. These are incoming signal. They are coming inside towards the brain or towards the spinal cord. Incoming. So this is sensory. We call them at sensory system or sensory division of the nervous system. There is something called as after the information reaches to the brain, brain will give the command to that organ through the nerves, and these nerves will carry the information to the organ. Sorry, to the organ. So they are going out, they are exiting. They are going out from the nervous system to the organ. We call it as efferent division. E for exit, E for efferent. Also, it is known as motor. Most of the time, it is causing the movement, contraction of the muscle, or secretion of the hormone, or the secretion from the gland. So they are called as the efferent. So the nerves which are giving out the information, taking the command to the organ, they are called as efferent nerves or efferent division of the nervous system. Even in the efferent system, there are nerves specifically controlling these activities, but under our own voluntary control. Some nerves, they are under our voluntary control. Some commands are under our voluntary control. We call them as somatic nervous system. It is a part of efferent division, efferent or motor. So those signals from the spinal cord, which is going to the muscle, but these muscles are which muscles? Skeletal muscle. These are skeletal muscles. So these muscles will be under our own voluntary control. Suppose I want to grab the pen. So my intention is to grab the pen. So I will do the movement of my arm muscle, my forearm muscle, and my small finger muscles, so that I can grab the pen. If I don't want, I will release it. So this is in my control. That's why it is called as voluntary system. It is called as voluntary system. Whereas something other than that is called as involuntary system. Can I control the food which is passing from esophagus to the stomach? Can I control? Although it is passing through a muscular tube, which is esophagus, and then the stomach. Although this is also a muscle, but these muscles are not in my control. They are not in my control. And that's why they are called as involuntary. Involuntary means they are not controlled by my will, not controlled by my own choice, by my will. Will means intention. I cannot intend to control them. This is done through an automatic system. This is done through an auto system. That's why it is called as autonomic nervous system, or short form is ANS. So up till now you have seen CNS, central nervous system, PNS, which is peripheral nervous system, and ANS, that is autonomic nervous system, which we will discuss detail in 2 lectures after these introduction to the nervous system. So autonomic nervous system is the system which is not in our control, usually controlling the internal organ of the body, Okay. So exit system or efferent system has got 2 parts. Efferent system has got 2 parts, which is somatic, supplying to the skeletal muscle, and autonomic, supplying to the controlling the involuntary system or controlling the smooth muscles usually. Again, autonomic nervous system is divided into 2. Autonomic ANS is divided into 2 systems, 2 parts. One is called as sympathetic, and another one is called as parasympathetic. Again, we will discuss it in detail in our next lecture. So what they are supplying? They are supplying, or they are controlling the cardiac muscle, the smooth muscles, and the glands. They are controlling the cardiac, smooth and glands. So this is the efferent division of the peripheral nervous system. Some authors say that there is one more system which is can be a part of autonomic nervous system. We call it as enteric nervous system. Enteron or enteric. What do you understand by enteron or enteric?
Skin?
No, this is related to gastrointestinal system, GIT. Enteron or enteric nervous system. It is the one which is controlling the contraction of the GIT. If you have gone through the structures, you will see that there are 4 layers everywhere in the GIT. If you cut open the organ, you will find there are 4 different layers, and in between these layers you will find the neurons or nervous system which are generating the impulse. They are called causing the contraction, and this is too much and too many neurons are there. So some people classify them as separate system, which is called as enteric nervous system. Although some other people say that this is a part of autonomic nervous system. So the one only responsible for controlling the gastrointestinal activity, it is called as enteric nervous system. This is also peripheral nervous system. Okay. Coming to the basic structural unit of the nervous system. Whenever we start to learn any organ, any system, we learn a basic structural unit of that, which is a cell. So cell related to the nervous system is known as neuron. What are the parts of the neuron? You already covered it.
Cell body, dendrite, axon, axon terminal,
Yes,
axon
yes.
hillock. Yeah.
Very good. And at the end, axon terminals will be there. We have seen in the previous class these axon terminals ending on the another neuron or maybe on the muscle. So we have got the basic structural unit of the nervous system as neuron. Now neuron could be of 2 or 3 different types. One neuron is specialized for, suppose this is a nervous central nervous system. Brain, we'll see this is a brain and this is a spinal cord. So some neurons will carry the information to the brain or to the spinal cord, bringing in. This is bringing in. These are called as afferent neurons. These are called as afferent neurons. Some neurons are specialized for taking the information to the organ, So they are exit the information. They are exiting the information or taking the information to the organ. They are called as efferent neurons. So neurons could be of 2 types, afferent or efferent, but it is not possible that they will be separate. There are some neurons which are in between them. These are called as interneuron. Interneuron. So neurons are of 3 types. One which is bringing the information to the nervous system is known as afferent neuron. The information which is taking from the nervous system to the organ is through the another type of neuron, another set of neuron, which is called as efferent neuron, which is taking the information to the periphery. Here it is towards the center, First is afferent is towards the center, and efferent are exiting or taking the information to the peripheral system. This is called as efferent neurons, and in between them is the some information is coming in, some information is going out, but there should be some connection in between, which is nothing but the interneuron. So this is the interneuron, afferent, efferent, and this is interneuron. You will you will find it amazing that number of interneurons are extremely high. Very high number of interneurons are there because in between connections are many. This is a diagram to show you what is mean by afferent and efferent. Afferent is the neuron which will carry the information toward the center, toward the nervous system. This taking the information here. From where it arises? It arises from a sensory organ or a receptor. It arises from a receptor. So sensory neurons starts with a receptor. Then they will take the information to the spinal cord or to the center or to the brain. And after that, some changes happen in their locally modified signal will come out through a neuron, which is called as efferent neuron. So efferent neurons are outgoing. They are outgoing information. Now you will say, what is mean by a nerve? This is a nerve because all the signals passes through the nerve from CNS to the organ, or from organ to the nerve, organ to the CNS. It is passing through a nerve. Nerve is nothing but collection of neuron. This is nothing but collection of neuron. Now collection could be only of one type, that is only sensory. So we call them sensory nerves. Those which are having only the fibers, only the neurons, which is of afferent type, we call it a sensory nerves. There are nerves which are carrying the information from the nerves, from the organ and from the CNS, both together, which will have, or only they are carrying, suppose there are some neurons which is only bringing the information from the nervous system towards the organ. We call it as motor nerve. Motor nerve. The nerve which is carrying the information towards the center, it is sensory nerve. The nerve which is carrying the information from CNS to the organ, it is called as motor or efferent nerve. And some are mixed nerve. Some are mixed. Mixed means they will have sensory as well as motor fibers. So here it is the description. Afferent neurons has sensory receptors. They will start from the sensory receptor and then they will bring the particular signal or stimulus toward the center. They will be the axon will carry the information towards the spinal cord, Whereas efferent neurons are also known as motor neurons. They will carry the information from the nervous system, from the central nervous system to the periphery. That's why they are called as efferent neuron, and they will be innervating either the gland or the muscles. They will be carrying the information to the glands or muscles. Interneurons are the neurons in between the afferent and efferent. So they may be forming so many connections because they are modifying the signal to the desired function. And you will amaze that out of all these neurons, 99% are interneurons. Only 1% are afferent and efferent separate. We have got around 100 billions of interneurons, which is found in the brain as well as in the spinal cord. Okay, apart from the actual performing cell, those responsible for carrying the information to the periphery or from the periphery to the center, there are some cells which are helping or supporting. These are called as glial cells. As they are related to the nervous system, we call them as neuroglia. Neuroglia, these are nothing but supporting cells. Supporting or accessory cells. What are these? These are nothing but the supporting. So they are mainly made up of connective tissue. Cells are the connective tissues of the central nervous system. Glial cells are the connective tissue or supporting cells of the central nervous system. what they will do? They will support the interneurons physically. Physically, they are supporting. Also, they are supporting it metabolically, maybe causing some changes in the chemical nature of that secretion or the neuron, and they are also helping it functionally. So these are nothing but cells which are responsible for physically supporting, maybe some are wrapping, maybe causing changes in the concentration of different ions metabolically, or sometimes they may be modifying the action of the neuron, or functionally they are responsible. So glial cells are responsible for physical support, metabolic support, and functional support. They are glial cells, they are not neurons, so they will not be conducting any information, any impulse. So impulse, or you can say the action potentials, it is only traveling through the neuron, not through the supporting or glial cells. There are 4 types. Basically, they are divided into basic types as number 4. 4 types of glial cells are there. What are these? The names are given as astrocyte, oligodendrocytes, microglia, and ependymal cells. These are the 4 major types of supporting cells, or or accessory cells, or glial cells. What are these? Astrocytes, oligodendrocytes, microglia, and ependymal cells. Everyone is doing specialized function. So this is just the diagrammatic representation of the nervous system, how these cells are helping or where they are located. So if you see around the cavities, you will see that in the brain is not a solid structure, but it has got some cavities. If you see, this is a cerebral cortex or cerebrum, and this is the spinal cord. If you open, you will find that there is some hollow space in the brain, as well as it will continue here also. So these are called as the ventricles. These are called as ventricles. Lateral ventricle, third ventricle, fourth ventricle. These are open, or you can say hollow space. These hollow spaces are having some cells lined to them. Here there are cells which are lined to them. These are nothing but the ependymal cells. Also, you will see that these are neurons. These are the neurons, one neuron and second neuron. Over the neuron, you will find that the axon is covered. This axon is covered by what? What is the covering of the axon?
Oligodendritis.
Name of the covering. What is this name of the covering? Myelin sheath. Very good. This is the myelin sheath. This myelin sheath is formed by a special cell in the nervous system. This is in the nervous system, or in the CNS, central nervous system. In the central nervous system, this myelin sheath is produced by a cell. We call it as oligodendrocyte. Myelin sheath is produced within the nervous system, within the central nervous system, through the cell which is called as oligodendrocyte. Whereas in the periphery, what is the source of myelin sheath in the peripheral neuron?
Schwann cells.
Very good, excellent. Schwann cells. Schwann cells are for peripheral nervous system, for peripheral nerves, where it will form the myelin sheath around the axon, Schwann cells. It is in the periphery, whereas oligodendrocyte in the central nervous system. One more cell which is found here, the pink or red color, I don't know how you are seeing. These cells, these are called as astrocyte. And you will see their one foot is on the blood cell, blood capillary, and another foot is on the neuron. They are putting their feet in the 2 areas, blood as well as on the neuron, which is responsible for making blood-brain barrier. Astrocytes are responsible for making a blood-brain barrier. That means if you see here, it is the blood, and here are the neurons. Anything which is present in the blood, let's say we eat some meal which is containing some chemicals, maybe having sodium, maybe having phosphate, maybe having any different chemical. If it is mixed with the neuron, then neuron function will be disturbed. Neuron function will be disturbed because we are eating a lot. Anything we are eating, we are not taking which one is responsible, or on the can or on the food, we are not taking the label. We are not reading the label which chemical is present. We are eating as it is. In our body, there is a system which will not allow these chemicals to reach to the neurons or to the nervous tissues, because nerves are nervous cells or nervous tissues are responsible for controlling everything. So if it is affected by these chemicals, then our brain will be disturbed every time. So astrocyte function as forming this barrier which is what you call barrier in Arabic, something which is preventing to mix.
حاجز doctor.
Hmm?
حاجز, I think.
Hajis. Okay. Okay. So anything which is like a wall, which is not allowing anything going from here from one room to another room. So this is a barrier is created by astrocyte, which is we call it as blood-brain barrier, BBB. So neurons are not affected by the content of brain, by the content of blood, because of a guard which is called as astrocyte. So astrocytes are creating the blood-brain barrier. Okay, but there are some microorganisms. There are some even some changes happen into the cells around the brain. So it has to be controlled. Like in our body, if some organism enter, there is an immune system which is causing formation of antibodies or defending it. So in the brain also there are immune cells. We call them as microglia. Microglia. So here are the functions of each cell. Astrocyte, formation of blood-brain barrier, plus it is also responsible for forming the scar tissue. Scar tissue. Whenever there is some damage to the brain, scar tissue is also formed by the astrocyte. It helps to maintain the ionic concentration around the neuron to the proper level. Ionic concentration around the neuron in a proper level, so that neuronal excitability will not be hampered or changed. So this is the function of astrocyte. As you said, oligodendrocyte are responsible for forming the myelin sheath covering of the axons in the central nervous system. In the peripheral nervous system, another cell is called as Schwann cell. Microglia, they are the immune cells of the nervous system. They are responsible for phagocytosis. What is phagocytosis?
It's the
Help in
exit.
defending.
Yes, this is a microglia. If some organism or any debris found here, then they will reach to this cell, to this area, and they will engulf it. And they will engulf and eat it, break down into smaller pieces, and then it will be eaten by the cells. This is called as phagocytosis. Phagocytosis, eating. They are eating anything which is harmful to the brain. Either it can be a bacteria or it can be a dead cell. Also, it will release nerve growth factor. Nerve growth factor for in case of damage or in case of in the initial stages to increase the size and the number of neurons, forming new neurons, which is at the beginning. but during damage, it is just helping it to increase the growth of a myelin sheath or damaged cell. Ependymal cells, as I said, they are lining the cavities. Suppose this is a cavity, so these are the cells, and they are secreting a liquid which is called as cerebrospinal fluid, CSF. Ependymal cells, they line the cavities like the lateral ventricle, third ventricle, fourth ventricle, and they will be secreting a substance, a liquid, we call it as cerebrospinal fluid. Also, they work as a stem cell. Ependymal cells, they also work as a stem cells. What is the meaning of stem cell? Stem cell means what?
The origin of cells.
Yes, the original cells which can modify, which can transform into different type of cell. The initial cell which can transform into the different type of cells, we call it as stem cell. So this is for forming the new neurons and glial cells. Forming new neurons in the glial cell, it is the function of ependymal cell. Okay, up till now we have seen how the nervous system is classified: central, peripheral, in the peripheral, sensory, motor, and autonomic. And then we have seen the different types of accessory or protective or supporting cells, which are called as the glial cells. There are 4 types: astrocytes, microglia, ependymal cells, and oligodendrocytes. Now we will move to the coverings, how the brain is protected, because any important structure in the body, it has to be protected to its proper level. The protection of the brain is by 2 means. First of all, the very delicate structure, it has to be kept in a cavity which is cavity which is covered by bones. So brain is covered by or placed in the skull. It is covered by the skull bone, so that it should not be damaged easily. This is one protection, that it is under or surrounded by the bones. So brain is covered by skull bones, whereas spinal cord is running through what? Through the vertebral column. If you remember the structure of a vertebral column, you will see that there is a central canal here. This is the vertebra. And here is the central canal. Through this, the spinal cord is passing. So it is covered by, or it is surrounded by the bones. Whereas around the brain also you will have very hard structure, which is the skull bone. This is the person. So the brain is protected under the skull bone, as well as the spinal cord is protected in the bony structure, which is called as central canal of the spinal cord. Oh, sorry. Not only the bone, but there is tough covering. There is membrane around the bone, around the brain, as well as the spinal cord. There is membrane, tough membrane is there, which is known as meninges, which are known as meninges, not in one, but in layer of 3. 3 layers of covering is there, which is called as meninges, which is lying between the bone and directly to the substance of the brain. Bone, as well as outside it is bone, and inside it is the the brain or the spinal cord. In between there are layers. These layers are known as the meninges, and they are classified. They are named as I said, there are 3 layers. So suppose this is here it is the bone, and here it is the brain, actual brain. In between there are 3 layers. So outer layer towards the bone is dura mater. Outer one is dura mater. Then in between is the arachnoid matter. and the innermost, which is sticking to the brain matter, is the pia matter. So dura, arachnoid, and pia matter. So now can you tell me how will you say this is the, suppose I will draw it in a horizontal line. This is dura, this is arachnoid, and this is pia matter. 3 layers are there. 3 layers of covering is there. What do you call this space? The area below the dura matter or under the dura matter? What do you call?
Cavity?
No, it is a cavity, but the specific name is there. Whenever suppose there is a boss, there is a boss is there, and under this boss there are 2-3 employees are there. We call them as subordinate. I am the boss and I have 2-3 persons below me. I call them as subordinate. So area below the dura matter, we call it as subdural space. Subdural space. Okay. Anything below, we call it as sub. This is arachnoid matter. Arachnoid layer of the covering. This is called as arachnoid matter. So what do you call this space now? The subdura matter is, subdural space is below the dura matter. So what is the name for any space below the arachnoid matter?
Space.
Arachnoid space. Very good, very good, very good. Subarachnoid space. You will see why I am telling you, or why I am stressing on these different terminologies, because you will see that CSF is present in this cerebrospinal fluid is present in subarachnoid space. See, this is area where you will see the liquid, which is called as CSF. Okay, so CSF is present in a space which is below, or you can say in between which and which layer? Arachnoid layer and pia matter of the meninges. Arachnoid and pia matter, these 2 layers, in between these 2 layers, there is some liquid which is called as subarachnoid space, and this the fluid is known as cerebrospinal fluid, and its function is to cushion. It's to cushion, make it soft, so that no vibration, no physical touch, or you can say no physical movement should occur. It's a sort of a cushion. So to avoid any damage to the which structure? Below this structure, that is called as the brain. So below the brain, or below this pia matter, is the brain, actual brain. So it is outside is bone. First is bone here. Then there are 3 tough coverings. One is dura, second is arachnoid, and some third one is pia matter. In between the arachnoid and pia matter, again there is one more protective layer, which is of fluid, which is called as cerebrospinal fluid. So this is how the brain is protected physically. Now also the brain is protected by one barrier. We call it as blood-brain barrier, where anything which is present in the brain, in the blood, cannot easily can enter into the brain and cause changes. So that's why there is some called as virtual barrier. It is not, you will not see them. You will not see like a layer. You will not see like a layer. This is blood and this is brain. You will not see them as physical barrier. This is blood and this is brain. You will not able to see clear cut layer, but there is some restriction of movement of ions or chemicals from the blood to the brain. And this is through a system, we call it as blood-brain barrier. Okay, so what we have covered is shown here. This is the skull where you will find the hairs. This is the scalp. Again, very tough skin layer. Then below the scalp is the bony skull. Below the bony skull, you will find 3 layers of membrane: dural, arachnoid, dura mater, arachnoid matter, and pia mater. The innermost layer is the pia mater, which is running along with the brain. In between the in between the dura and the arachnoid is the subdural space. Below the arachnoid and above the pia mater is the subarachnoid space. This is the subarachnoid space where fluid is present, which is known as the cerebrospinal fluid. This is all over, all around the brain. It is there, all over. Sorry. It is present all over. You will see in around the brain as well as around the spinal cord. It is going around the spinal cord. These arrows will show that this fluid is around the spinal cord also. CSF is present around the brain, acting as a shock absorber, and around the spinal cord. Okay, in between the 2 layers, that is dura between arachnoid and the pia mater, between arachnoid and the pia mater. How much is the cerebrospinal fluid? Cerebrospinal fluid is total around the brain and the spinal cord at any given moment. It is about 125 to 150 ml. 125 to 150 ml. It is present in the subarachnoid space around the brain as well as in the spinal cord. It is formed by the choroid plexus or capillaries in the ventricle of the brain. In the ventricle, I told you that brain is not a solid structure. It has got some central hollow areas. These hollow areas are known as ventricles. So if you cut open the brain, you will find some space here in the center. This is called as ventricle, and this ventricle is connected to the space below also. So lateral ventricle, third ventricle, and fourth ventricle. It will be placed here also, some subspaces here. So total about 125 to 150 ml of the fluid is present around the brain and the spinal cord. This is cerebrospinal fluid. Okay. This is forming through the capillaries. The cerebrospinal fluid is from where it is coming? It is coming through the capillaries. Capillaries are forming, and capillaries contain what? Blood. So it is formed by the blood only. And it is absorbed again in the venous system. It is absorbed. It is not that it is formed and it will be kept like that. It's not only 125 ml which is present since birth. It's not like that. It is secreted and it is absorbed. So at any given moment, it will be around 120 to 150 ml. So there is continuous circulation is going on. Continuous circulation, forming, absorbing, forming, absorbing, forming, absorbing. But at any given moment, if you calculate them or if you know the quantity, it is about 125 to 150 ml. If this formulation is there and absorption is stopped, only forming, what will happen to this quantity? It will remain 150 ml, or it will increase, or it will decrease. What will happen? It will increase. So increase in this fluid formation will lead to a condition we call it as hydrocephalus. Hydrocephalus is increased amount of cerebrospinal fluid. What are the causes? Causes could be increased formation, or causes could be decreased absorption or blocked absorption, so that it is accumulated only around the brain. Okay. So two conditions are there. Either it will be secreted more or it will be absorbed less, or there is a block in the circulation. We call it as hydrocephalus, or in general term, water in the brain. So if it is not treated, if this is becoming more, let's say 200 ml or 250 or 300, what will happen? It will compress because this is around the brain. Yeah, it is around the brain. So it will compress the brain. It will make pressure from each side or a specific side. So lead to damage to the brain and mental retardation. Damage to the brain or mental retardation. It is usually a most of the time it is a condition which is present by birth. Immediately you will observe that the baby's head is very big. Why? Because all around is the water accumulated, or CSF is accumulated because of excessive secretion. And babies' bones, skull bones are fragile and they are not connected, so it will become bigger and it will cause difficulty in delivery of the baby at the time of the birth. After the birth, what the doctors do, doctors will find out the cause why the increased amount of CSF is there. So either suppose the fluid is forming here, CSF is forming here, and there are veins, venous system is there. So what they will do, they will make a shunt. They will make a connection by surgical operation. They will make a passage between subarachnoid space and a venous drainage, venous area, maybe in the jugular vein or maybe any vein which is around it. So this is shunting, shifting the fluid from formation area, that is the subarachnoid space, to the venous system. This is called a shunt. So you can decrease the pressure by draining that into a venous system. So this is all about the lecture number 1. Some important points other than what we have just seen, that brain function depends upon two things: supply of energy and supply of oxygen. Oxygen supply should be continuous, and energy in the form of pure glucose. In the form of glucose, not other proteins or something. It requires continuously. So oxygen plus glucose are required all the time continuously to function properly. If there is stoppage of oxygenation, maybe you stop your breathing for maybe around 4 to 5 minutes, the brain will get damaged. Also, if the glucose supply is not there or hampered or stopped for 10 to 15 minutes, again the brain will get damaged. If the glucose is decreased, not completely finished, if glucose is decreased, what will happen? Sorry. If glucose supply to the brain is decreased, what is happening to the brain? Brain will get irritated.
Will be damaged.
Not damaged, but it will start irritating. Irritated. Will function abnormal first, which is usually seen after a long fast. When you are fasting, now in a month of Ramadan, maybe 8 o'clock in the morning, 8 a.m., you will be okay. 10 a.m., again okay. But what is happening after 5 a.m., 5 p.m.? All will become crazy. If you are traveling, if you are in the road, you will be, if someone crosses you, you will start to shout. Why? Because your brain is lack of glucose and it becomes irritated. It will not take proper decision. It will not take proper decision. So glucose, what you call this decrease in the glucose? Hypoglycemia. Hypoglycemia will lead to irritation of the brain and will become crazy. Thank you very much for attending the lecture number whatever 12.
Thank you, doctor.
Question.
Yes. Do you have any question?
Yes, we said
Yes.
that the cerebrospinal fluid is formed from ependymal cells, but then later
Yes.
we all that it is formed with some other cells, the choroid plexus.
Yes. Choroid plexus is formed where? Choroid
In the
plexus
ventricle.
is nothing but the neuron, the what you call here. So these are the ependymal cells and they are lining the cavities. And these cavities are also receiving capillaries. All of them will lead to form the choroid plexus. Choroid plexus is nothing but the combination of capillaries and ependymal cells because the cells require blood. Basically, blood is required. So blood is coming through the capillaries and it is supplied to the ependymal cells and they will convert the blood into CSF. So basically, CSF is formed by the ependymal cells with the help of blood. Agreed? Or you have still doubt?
So, what is the choroid plexus?
What is plexus means? Plexus.
Collection of.
Brachial plexus. Have you heard about brachial plexus? Zigzag, crisscross. So for the nerves, it is called as brachial plexus where it is supplying the nervous supply to the arm. If it is a crisscrossing or intermingling of the capillaries, which is present in the ventricular cavities of the brain, along with the lining of the ependymal cell, we call it as choroid plexus. Agreed
Okay.
or still? Yeah, still doubt. Literal meaning of plexus is crisscrossing or intermingling. Choroid plexus. It is formed by the choroid plexus, which is nothing but capillaries in the pia mater of the ventricles in the brain. So capillaries, along with the ependymal cells lining the cavities, will be helping to convert the plasma into CSF. So basically, if you take CSF and do the analysis, it will be similar or it is having similar content as of the plasma. One of the tests for knowing whether there is a brain is infected or not, or if the person is, you can say comatose, what do? You take a sample for analysis from the brain in the form of cerebrospinal fluid. In the form of cerebrospinal fluid, and this sample is taken through the subarachnoid space, which is present all over from the spinal cord as well as the brain. So we will not put the needle in the brain. What we will put? We will put a needle in the vertebral column. In the vertebral column, these are the vertebra. And here is the spinal cord. This is outside, huh? This is toward the center. So we will put a needle here. And in the subarachnoid space, we will take some drops of liquid or CSF. CSF in between the two vertebra at the lumbar area. Lumbar, third and fourth vertebra. In the lumbar region, suppose the person's back is like this. This is the back of the person. I will show this is the back of the person. These are the arms, the legs. So we will put a needle here in between L3 and L4 to bring the CSF, and we will test it in the lab for finding out what is the cause of the disease related to the brain. So this is called as CSF analysis. CSF analysis, and this procedure is called as lumbar puncture. Lumbar puncture. Do the analysis of the brain, analysis of the fluid which is around the brain as well as around the spinal cord. Lumbar puncture. This is a procedure called as lumbar puncture. Please check on the internet what is the use. Lumbar puncture. So lumbar puncture is to get the CSF and do the analysis on the CSF for any disease related to the CNS or either central or peripheral nervous system. But if the person is comatose, person is having some consciousness problem or suffering from any infection in the brain, we do CSF analysis, and it is taken by a procedure called as lumbar puncture. Okay, any other question? I hope you understood now.
Yes, sir. Thank you.
Any other question?
Doctor,
Yes.
do you take attendance from the
From the those who are participating in the meeting, I can take the attendance from there.
yes,
Now
but
here,
okay.
yes, I will take it. It will be automatically recorded because you people join with the college ID, right? University ID at UM.
Yeah, unfortunately, I didn't. I just realized. Could I
Okay.
send you my my ID?
Okay, what's your name? Your SA coming. Name is coming as SA.
Yes, my name is Sarah. Hazza.
Sarah. Okay, Sarah. I will
Okay.
write it down here. I do have a pencil.
Would you like me to give you my ID number?
Yes, please. Sure.
Okay, 23
23
122
122
00
00
36.
36. Okay,
Thank you, Doctor.
Sarah. Yeah, welcome. Anyone who can you see the chat? Is the chat is available? Because sometimes there is a problem here in the chat. So if anyone wants to send some number message, you can send it through the chat or maybe directly through your group leader to me. No problem, through WhatsApp.
Doctor, should we study lumbar puncture?
This is something extra.
All right, thank you.
Not for the exam.
Yeah, clear.
Good. So we will take a break of 5 minutes so that wherever you are sitting, standing, so change your position, move around in the room or place so that we will start the second lecture. So second lecture we will continue. You don't have to log out. Just remain here. I will stop for sharing for this slide and we will start the next lecture within 5 minutes. Now it is 11:39, 11:45 we will start second lecture, inshallah. Let me stop the recording and make it another one so that 2 lectures will be separate.